Hello friends, welcome to another lecture in botany. Today our topic is Stelar Evolution in pteridophytes. First, we will try to understand the stele and its different types, and then we will discuss its evolution. The name stele has been derived from Greek word meaning pillar or column. Sachs in 1875 proposed that the vascular system of the plant body is a continuous system. For the first time, the stellar organization appeared in pteridophytes. Hence, the pteridophytes are called as first vascular plants. The stellar theory was proposed by Van Tighem and Dolit in 1886, according to which the root and stem are fundamentally similar in gross anatomy because in both the cortex encloses the central part of the axis called the stele. According to them, stele is the core of the axis, which includes the vascular system, interfascicular portion, pith if present, and some surrounding portion of the fundamental tissue in the vicinity of vascular bundles. The term stele is applied to the primary tissue only. On the basis of structural variation in the primary vascular system, following types of steles have been recognized. Number 1, protostele, Siphonosteles, Solenostele, Dictyostele, Eustele, atactostele, and polycyclic stele. Let us discuss them one by one. Protostele. This is the simplest type of stele. It consists of a solid central column of vascular tissue. Pith is absent. Xylem is located in the center, surrounded by phloem. On the basis of shape of the xylem, following types of protosteles have been recognized. Number 1, haplostele. This type of protostele has a smooth core of xylem surrounded by a uniform layer of phloem. It is found in Lycopodium syrenum and Selaginella cruciana. Actinostele. This type of protostele has a star-shaped xylem core. Here, phloem is present in the form of separate piths, alternating with the projecting parts of xylem. It is found in Lycopodium serratum and Xylotum triquetrum. Plectostele. In this type of protostele, xylem occurs in the form of separate plates which lie parallel to one another with phloem situated between them. It is found in Lycopodium volubile and Lycopodium clavatum. Mixed protostele with phloem. In this stele, xylem groups are scattered in the form of irregular patches that are embedded in the ground mass of phloem. It is found in Lycopodium syrenum. Mixed protostele with parenchyma. In this stele, xylem groups are scattered in the form of irregular patches that are embedded in the ground mass of parenchyma. It is found in Hymenophyllum demissum. Now the Siphonosteles or medullated protostele. In this type of stele, there is present a pith. Here, the vascular tissue is arranged in the form of a hollow cylinder with distant pith in the center. It is found in the stems of most members of Phyllocophyta. It is of the following two types. Ectofloic Siphonostele. It is a type of siphonostele where xylem cylinder lies next to the pith and is surrounded by the phloem cylinder on the outer side only. It is found in Osmunda and Equistum. Amphiploic Siphonostele. In this type of siphonostele, the pith is surrounded by inner endodermis, inner pericycle, inner phloem, xylem, outer phloem, outer pericycle, and outer endodermis. So, in this case, phloem surrounds the xylem internally as well as externally. It is found in Hediantum and Marsilia. In both ectophyloic and amphiploic siphonosteles, vascular tissue occurs as continuous cylinder. This is because leaf traces do not break the vascular cylinder. Such plants in which the vascular supply to the leaf is without any break in vascular cylinder are called microphyllous. Now the solenostele. This is similar to siphonostele in having central pith, but differs in producing leaf gapes whenever leaf traces originate. Thus, due to production of leaf gapes, the cylinder becomes dissected at places. This type of solenostele may be ectophyloic or amphiploic depending upon the type of siphonostele from which it is produced. Such plants where leaf gapes occur in vascular cylinder are called megaphyllous. Next type of stele is dictyostele. In many siphonostelic Phyllocophyta, leaves are inserted on the stem in close succession. In such cases, leaf gapes overlap in their longitudinal extent to such a degree that vascular cylinder of the stem appears dissected into tubular network of interconnected longitudinal strands called meristeles, separated from one another by leaf gapes. These meristeles in a transverse section appear arranged in a ring. Such a stele is known as dissected siphonostele or dictyostele. In a dictyostele, each meristele has a general structure of protostele. It is found in some species of Dryopteris. Eustele. This is a modification of siphonostele in which vascular system consists of a ring of collateral or bicollateral vascular bundles. These are separated from one another by wide medullary or interfascicular regions, and the leaf gapes are not clearly distinguishable. It is found in the stems of Gymnosperms and Angiosperms. Atactostele. This is the most complex type of stele. In this type, the vascular bundles are irregularly dispersed in the ground tissue as in the stems of Monocotyledonous. Polycyclic stele. In this case, more than one steles are present in the axis of pteridophytes. For example, 2 in Selaginella cruciana and 16 in Selaginella levigata. Now the Stelar evolution. Jeffrey in 1898 proposed that protostele is the most primitive type of stele from a phylogenetic standpoint, from which other types of steles have evolved in the course of evolutionary specialization. It is considered to be fundamental stellar organization that was present in the earliest vascular plants and is now retained by some living vascular cryptogames like Xylotum, Mesopteris, and Lycopodium. The primitive vascular plants, like the extinct Xylophytales, also possessed protostelic vascular organization. Presence of exclusively protostelic stems in the earliest vascular plants and their retention in some of the living vascular cryptogames lends a strong support to Jaffery's view that protostele is a phylogenetically a primitive type of vasculature. In its simplest form, protostele is haplostelic. During further elaboration, the central core of xylem became irregular and assumed almost a star-like shape. Such a modification of stele is termed as actinostele. As a result of further evolution, the xylem splits up into a number of parallel plates with phloem. Such a modification is called plectostele. It is found in some species of Lycopodium. Haplostele to actinostele and then to plectostele is considered to be one line of evolution of the protostele. It is regarded as Lycopsid line of evolution. Another very important evolutionary change that occurred in protostele was the presence of the central pith. This step led to many important changes in the protostelic organization and gave rise to complicated stellar types. Two theories have been proposed accounting the phylogenetic origin of the pith. These are intrastellar theory and extrastellar theory. According to intrastellar origin of pith or expansion theory, the inner vascular tissue metamorphosed into the parenchymatous pith. The occurrence of mixed pith in some living forms supports this view. A mixed pith shows trichoids within the parenchymatous pith. This may be looked upon as a transition stage between a true protostele and true Siphonostele. According to the extrastellar origin of pith or invasion theory, the pith is cortical in origin. The parenchymatous cortex is said to have invaded through the leaf gaps and branch gaps into the center of vascular cylinder to give rise to the pith. But this cortical invasion could not have produced polycyclic Siphonostele. Ames in 1936 considered that in primitive forms it may be intrastellar in origin and in high forms extrastellar in origin. Appearance of pith led to the conversion of the protostele into a new type of stele called the Siphonostele. Elaboration of Siphonostele also followed two courses of evolution. The appearance of pith resulted in the formation of a stele consisting of a central pith surrounded by a complete wing of xylem, which in turn was surrounded by a complete wing of outer phloem, pericycle, and endodermis. Such a stele was named as ectophloic Siphonostele. In its simplest form, such a stele is uninterrupted by leaf gaps and is called cladosiphonic. In the megaphilous vascular plants, the complete and uninterrupted cylinder of ectophloic Siphonostele became interrupted by the appearance of leaf gaps, which is now called phylosiphonic. In case the leaf gaps do not overlap, the stele is interrupted only at considerable distances or nodes by a one leaf gap, so in between the two leaf gaps the vascular cylinder remains complete. Such a stele is called Siphonosteles. During the course of evolution, in certain pteridophytes, leaf gaps on the stem overlapped and led to the formation of a much dissected stellar organization called Eustele. It is made up of a number of separate and collateral vascular bundles. In certain cases, the vascular bundles are scattered as in monocotyledons. The stele was termed as atactostele. During the another line of evolution, the modulation of protostele was followed by the appearance of phloem on either side of the xylem. Likewise, internal pericycle also appeared. As a result, the Siphonosteles consisted of a central pith surrounded by internal endodermis, internal pericycle, internal phloem, external pericycle and external endodermis. Such a stele led to the formation of a amphiphloic Siphonosteles. It may be cladosiphonic or phyllosiphonic. The phyllosiphonic amphiphloic Siphonosteles with only one leaf gap at the node formed amphiphloic Siphonosteles. In case the leaf gaps overlap, the resultant stele is called the dictyostele. Dictyostele is very common in Phelicophyta. In many eusporangiate and leptosporangiate ferns, the dictyostelic stems are protostelic at their bases. Recent experimental studies also reveal that dictyostelic condition can be changed to solenostelic or even protostelic condition by removing the young leaf primordia from shoot apices. All these observations prove that protostele is the basic or the fundamental stelar type from which the complicated steles or vascular systems arose by elaboration. Among the pteridophytes, polycyclic stele is the most advanced condition exhibited by some ferns like Maracea and Matunia. It also originated from the protostelic condition by further elaboration. This is borne out by the fact that in Matunia pectinata, there is a regular transition from protostelic condition to solenostele and then to polycyclic condition. This developmental phenomena is termed as recapitulation. Occurrence of such a developmental phenomena leads further support to the Jeffreys view that protostele is a primitive condition. Thus, the evolution of stellar organization have taken place along several independent lines. Even a single genus like Galicinia shows some species with protostelic structure and some with siphonostelic organization. So, the stellar structure has not of much significance in establishing phylogenetic relationships. So friends, you might have understood the concept of stele and its evolution. See you with a new topic. Till then, goodbye.